This is going to be the first time the science guys lighten it up. So, Catherine, that was great. Rajat, super. But you guys are going to be late to your first meeting, so I'm going to be very quick. Tomorrow, you can hear me speak again in regard to what we're doing in the science realm regarding research. But I'm briefly going to let you know that what we're chasing right now is the following. We're continuing to push research into basic discovery of ideas. Those ideas are the fruit of the future. The challenge that we are going to be faced with, and what I'm trying to do with our translation program, is move the ideas into medicine. And then, with those medicines, essentially test them in the clinic to see if the medicines are efficacious. So it's a 3-part song that I hope you guys continue to sing with me. Move the ideas, discovery to ideas, ideas to medicine, medicine to essentially practice. So that's what we actually set this meeting up around. We had a basic science discovery meeting yesterday morning, a translational meeting that you'll hear from Dr. Katz on, and then there was a neurohabilitation meeting this morning that is essentially to help give our girls abilities that they haven't had. And then finally, we closed with a clinical session that was chaired by Alan. Now, you'll hear from Pam Diner tomorrow. She'll be talking about the neurohabilitation meeting and what it is they achieved there. Very quickly, on the basic science meeting that Sasha Nelson chaired, and I'm speaking for Sasha, new discoveries on what MECP2 does, and you're all saying it can't do more stuff. Yes, it can. So it's doing more. There's new, essentially forward-looking ideas with progenitor stem cells and how to manipulate stem cells so we can do high-throughput screening of drugs in the lab, which will be very efficient for essentially looking at different drugs and possibly tracking what those compounds are going to do to the cells when we actually go to the animal models. And then some very sophisticated neurophysiology was talked about by a number of colleagues, both looking at excitatory pathways and inhibitory pathways to try to understand how each of those pathways help us understand what's happening in your daughters. And finally, there was a very, very nice talk on astrocytes and how astrocytes are involved in the disorder. And lastly, a talk on MECP2 duplication and trying to understand some of what we can learn from that disorder and bring back into our house to help us in our house. So, David Katz is going to speak to you about the translation program. And the translation program is, remember, how do we actually move the ideas into medicine? And David is probably one of the foremost experts in the country in regard to Rett in this regard. So David, he has a plane in a little bit, so he's going to be quick as well.
Thank you, Steve, and good evening, everyone. As Steve said, translation is where we take ideas, discoveries about the basic biology of Rett syndrome and try and move those ideas into medicines using mouse models. Now, in order for that to happen, in order for ideas to become medicines and for those medicines to become treatments, requires a partnership. Any one laboratory is a partnership among scientists, but in order for that laboratory to move ideas to medicines, we need to partner with foundations like RettSyndrome.org, who bring us not only support, the research support to do the work, but also bring to us the human face of the disorder. And I think it's absolutely critical that scientists meet families and meet the kids so that we never forget who we're working for. So the advocacy organizations like RettSyndrome.org are part of the partnership. Secondly is the National Institutes of Health, who provide funding and the regulatory framework within which we do our research. And then the other key element is the pharmaceutical industry. Pharmaceutical companies and biotech companies who have the resources, both in terms of what drugs they've discovered, but also the financial resources that will ultimately be needed to run clinical trials. And so the session that we had yesterday on translation brought together all of these factors. We heard from the NIH about what they would like to see coming out of our laboratories in terms of promising ideas that they would then be interested in funding to turn ideas into medicine. We heard from the pharmaceutical industry as to what they need to see in order for them to consider a potential medicine to be something that they would actually be interested in investing in and taking all the way into the clinic. And then we heard from basic scientists who are creating that bridge between ideas and medicines. And so just very, very briefly, we heard, and this is an emerging theme now, we heard several examples from several laboratories that we are finding more and more ways in the mouse models to reverse symptoms of Rett syndrome. That is, we are discovering more and more pathways in the brain that we can manipulate, and at least in the mouse models, reverse symptoms. Now, that sounds really promising, right? And we're all very excited about that, but I want to manage expectations. Many things have been cured in mice, in other disorders, that have not translated to humans. So now that we are making this progress and discovering that we can reverse symptoms in mice, the challenge for all of us, including RettSyndrome.org, is to identify, and that's why we do what we do, is to identify what are the most promising new medicines coming out of the mouse studies, which have the best chance of translating into a successful treatment in patients. And that's largely where we're at now, is looking at the landscape, looking at what's being discovered in the mouse models, and through programs like the Scout Therapeutic programs that Steve created within RettSyndrome.org, using programs like that to pick the most promising candidates and gather the data that will give us confidence that we should not move something forward to clinical trials. So that's where we're at now, and I hope next year we'll have even more exciting news to share. Thank you.
So in the final session was our clinical research session. And before I bring Alan up, I want you all, for those of you who thought about or think back to 2 years ago at the World Congress in New Orleans, those of you who were there, Larry Glass got up on the stage during one of the evening events and said, "I'm in." And Alan is going to tell you about where that study is. So it was only 2 years ago that Larry Glass got up and said, "I'm in." And Alan, the floor is yours.
I have a flood up here. Good evening. So this is a very exciting day for me personally and for all of the clinicians who've been engaged in clinical research, because I think we saw really for the first time that we covered the waterfront yesterday and today, from basic science all the way up to clinical trials. We learned about, from an expert of clinical trial design, how one could go about designing clinical trials and different mechanisms that one could use. And believe me, this is not a trivial pursuit. This is very, very hard work. Now, in terms of concept, basic science is tough, but in terms of conduct, clinical trial management is really tough. So it's a tough field we're looking at. And having said that, we spent the first half of the afternoon talking about 3 clinical trials that are going on currently. 2 are with the compound or related fragment of IGF-1. The compound that Steve mentioned and Larry Glass is the tripeptide of IGF-1. That is in trial in Houston and in Birmingham and in St. Paul, Minnesota. It involves girls or women 16 to 45. And we learned that today they made the press release that the trial has actually completed enrollment and will be ending in early September. Thereafter, data will be analyzed and the decision will be made whether or not it's a no-go or go signal following meetings with the FDA. We also learned about the full-length IGF-1 trial, which is being conducted in Boston Children's. It is currently in a double-blind placebo crossover study, and that is a much longer study and will not be concluded for perhaps a year. But that study has just about completed enrollment. We also learned about a trial of an agent which is approved for use in other neurologic disorders and is being trialed at Montefiore Hospital and Albert Einstein Medical Center. It is called Copaxone, and it is being trialed in 10 girls, a bit older, 10 and older, and they've completed enrollment and study of 10, and they have 2 additional children that they're completing. These results are not available, not at any time, but all we can say is that the agents are safe. They have no side effects that have been noted yet, and they have been tolerated very, very well. We then concluded the afternoon session by looking at issues related to clinical trials. How does one select patients in a fashion that assures that you are really studying apples and not apples and oranges? So we call that patient stratification, and that's a bit of a difficult challenge, but something we're working toward. And then we heard about how one goes about conducting studies that have good outcome measures. We lack really good outcome measures now, but we are working on it currently, and we have results that are very, very positive, and I'm sure there will be some evolution of these over the next year or 2, but we have the basic framework in hand. I started out the session this afternoon by making a statement, and I'll end with that. Many of you in this room have never had the privilege of meeting Andy Rett or Bengt Hagberg, and Bengt is the person who wrote or championed the Rett syndrome in the English language in 1983, and it became a worldwide name and disorder. And my feeling was, or my impression after listening to the first day and a half, was how they would have felt about being at the meeting today. And I think a smile would have been on their face. So thank you.